Bismillahirrahmanirrahim. Today I would like to share the knowledge about Diptera I Ceratopogonidae and other name is culicoides or midges. My name is Sarfaz Rahman. I am a PhD scholar in the Department of Parasitology. I would like to share this knowledge with references these books: Veterinary Parasitology by second edition by G.M. Oropouche, Diagnostic Parasitology for Veterinary Technicians 5th edition by Charles M. Hendricks, Handbook of Entomological Terms by Muhammad Ashfaq, a Handbook of Veterinary Parasitology, Domestic Animals of North America by Hendricks J. Griffith, Medical and Veterinary Entomology, 3rd edition by Gary R. Mullen, Economic and Ecological Significance of Arthropods in Diversified Ecosystem by Chakravarti. Please subscribe my channel and also push the button of bell icon. So biting midges or other name is Ceratopogonidae, and these are the minute blood-sucking flies. You can observe in this picture, and you can see in this picture also. And commonly known as no-see-ums, their small size and the fact often go unnoticed despite their discomforting bites. Another name, punkies. In United States, the Dutch corruption of the Algonquian, the Native American, and the root punqua. Ash-like, its appearance of the fly as its biting, burning sensation like hot ash from a fire in contact with the skin. Early French Canadians called the brulette, and from the bruler meaning to burn, and it is also called the sandflies, particularly in the coastal areas of the South United States, the West Indies, and adjacent part of the Caribbean. Taxonomy of Ceratopogonidae: approximately 123 genera and 6 to 7 extant described species. Ceratopogonids are divided into 4 subfamilies: Leptoconopini, Forcipomaini, Daschalini, and Ceratopogonini. The species in only the 4 genera are known to attack the humans and other animals. Most important genus in this respect is culicoides. The principal vector that the BTV, that is the abbreviation of the blue tongue virus, and epizootic hemorrhagic disease in the North American ruminants, and this the C. sonorensis and C. variapenis. And you see in these two pictures, and this is the blue tongue disease, and these are also the Ceratopogonidae or the midges. Morphology of Ceratopogonidae. The Ceratopogonidae larvae are long and slender. You can see in this structure that is the long and slender, and ranging from 2 to 5 millimeter in length when mature, and the body is white color, translucent appearance in contrast to the yellow to brown head capsule. Thorax marked by a characteristic pattern of subcutaneous pigmentation. Thoracic and abdominal segments are similar in size, contributing to their elongate and cylindrical body shape. And you can see this the body shape is the cylindrical. So the anal morphology of the Ceratopogonidae, that the anal papillae a pair of the narrow bifid and function in osmoregulation. You can see in this picture, larvae lack the spiracles and depend upon the cutaneous respiration. Culicoides and leptoconaps larvae lack the thoracic and abdominal appendages, and Forcipomia and Atricopogoni larvae are well developed, and ventral, prothoracic, proleg, and associated apical hooklets or the CT. So the mouth parts characterized by a pair of mandibles that are not opposable. They move vertically or partially rotate while the larvae feeds used to scrape, tear or seize items depending on the species, and pupa that is brownish in color with the pair of the relatively short but conspicuous prosthoracic respiratory rising at the interior end. So the morphology of the Ceratopogonidae, the adult culicoides midges, these are tiny, usually 1 to 2.5 millimeter in the body length, mouth parts adapted for biting or piercing tissues, and well developed in blood-sucking species. In females, the mouth parts are surrounded by a prominent extension of that labium, and that the called as a proboscis, which is relatively short, about as long as the head, and consists of an upper labrum epipharynx and a pair of black blade-like mandibles, a pair of lacini, that is maxillae, and ventral hypopharynx, that the pairing median longitudinal groove along which the saliva is passed as the female feeds. Mandibles. These mandibles bear a row of the teeth along the inner edge near the tip, which is used to lacerate the skin while biting. The mouth parts of the males are generally reduced and are not used in blood feeding. And you observe all the structure of the mouth parts in this picture. So the life history of the Ceratopogonidae, the adult females typically require a blood meal to develop their eggs. Autogenous, carry over enough nutrients from the larval stage to developmental one, and the first gonotrophic cycle without feeding on the blood. Development of the eggs and requires 7 to 10 days, but may be as short as 2 to 3 days. Eggs are deposited in the batches on the that the number of the eggs per female varies from 30 to 450 or more, depending on the species and the size of the blood meal. Autogenous females tend to produce fewer eggs. The eggs are small and elongated, 250 to 500 millimeter in length. These are often like the banana shaped, and are covered with the minute projections. Functions in the plastron, respiration, white when first deposited, but gradually turn brown. Eggs hatches in around about 27 days. Larvae develop through 4 instars with a development time that varies from 2 weeks to more than a multiple oviposition cycles by individuals. You can observe in the structure, this is the life history or the life cycle of that Ceratopogonidae or the midges. So the life history of the Ceratopogonidae, the some species are abundant only in the spring. The culicoides are bigotus and C. niger, C. trovesi are present in high spring population. The C. spinosus abundant throughout the spring and summer and fall in C. crepuscularis, C. furens, C. hematopotus, C. stellifer, and C. venustus. Still others are bivoltine, where the peak in the spring and fall. And you observe in these two pictures, these are the different types of the ceratopogonides. So the behavior and ecology, ceratopogonids larvae develop in a wide range of the aquatic and semi-aquatic habitat in biomes ranging from the tropics to the Arctic tundra, and leptoconops species occur primarily in sandy or clay-like, you can see and you observe in this picture are the leptoconop species occur primarily in the sandy or clay-like and alkaline soil of the arid region, along tidal margins and coastal marshes, beaches, and Forsythomia, that species that found in mosses and algae in shallow water in more terrestrial habitats such as rotting woods. Larval habitat of the astroconops in is wet soil and detritus, and it is difficult to make the generalization about the breeding sites of the culicoid species. So the behavior and ecology, the Ceratopogonidae larvae often can be recognized by that the location and consisting of the side-to-side lash movement of the body, and you observe in this picture, and they propel themselves through the water or moist organic substrate, and culicoides larvae that generally consist to be good swimmers, especially the later instars. So the public health importance, the biting midges that are the source of annoyance caused by their persistent bitings, problems in coastal areas where the salt marsh species are notorious pests, often creating the great discomfort for the local or resident or head goers, hitch goers, and discouraging the tourism during the summer month. In the United States, the C. fiorense, C. hollensis, C. mulius are the most troublesome species, taking the humans and other Atlantic coast area. The culicoides mississippi piensis is a problem for almost in the Gulf Coast, and C. barbosae is commonly a problem near the mangrove forest of the southern Florida. Certain leptonoco species are especially the pestiferous along the coastal beaches, linili along the Gulf Coast, and the Bicoarity that is in the Caribbean, where the member of the L. culticeae group are the annoying biters in semi-arid regions of the southern United States. A common pest that throughout much of the eastern United States is the C. Paraensis, a tree-hole species that readily bites the human, especially during the late afternoon and early evening, causing the considerable discomfort to hunters, campers, and hikers, annoyance to the homeo owners while the picnicking or trying to work in their yards bordering deciduous woods. And you observe in this picture that this is a picture, and you also understand that how it is creating the discomfort. So Oropouche fever, and caused by a virus in the family Peribonea viridi, first isolated from the charcoal worker in the Trinidad in 1955. Virus has been documented in the the numerous epidemic in the of the Brazil region, and outbreak before 1980 were largely restricted to parasitic, and while we are over the 165,000 human cases of this disease were estimated to have occurred between 1961 to 1980, and since that time the outbreak have been reported in Brazilian state of the Amazon, the Goiás and Maranhão, and in the Amapá territory. Epidemics have in the urban areas where 44% of the local populations have been seropositive for this virus. Signs and symptoms: cause a non-fatal acute febrile illness, general muscular joint pains usually lasting 2 to 5 days, and more than half of the cases involve symptoms such as headaches, dizziness, photophobia, and severe malagia, arthralgia, which can occasionally lead to prostration, and rare occurrence of the symptoms often prolong the illness up to 2 weeks. Incubation period for this disease is believed to 4 to 8 days, and you can get the sketch from this picture. Other viral agents: many other arboviruses have been isolated from the culicoid adults, and 4 of them are members of that: Bania oviridi, Caribbean Congo, Rift Valley fever, Dubai, and Shuni virus. 3 mosquito-borne viruses: Eastern equine encephalitis, Japanese bee encephalitis, and Venezuelan equine encephalitis have been isolated from the culicoides and Lacheiolia species. There is no evidence, however, to indicate that biting midges' role in transmitting the viruses. And this is the picture. You can get the image. So the Mansonillosis, 3 filarial nematode species in the genus Mansonilla called the infestation in the humans called the Mansonillosis. Cases occur widely throughout the tropical and subtropical region of the both Old World and New World. Culicoid and Forcipomia and Leptoconops species served as arthropod vectors, and although infestations involving these parasites are generally mild or asymptomatic, cause serious medical problems. And this is the picture of the Mansonillosis. So, veterinary importance: the biting midges serve as the vector of more than 35 arboviruses that infect the domestic animals. Only a few of these viruses cause significant clinical diseases. Cattle, sheep, horses, usually are other like animals. Most of these viral agents are members of the Rheoviridae or Buniaviridae, including the pathogens that causing the blue tongue disease, epizootic hemorrhagic disease, African horse sickness. The 2 other family of the virus of which culicoid species are having the implicated as a vector are the Rhabdoviridae and Poxviridae. For most of these viral agents, the principal Ceratopogonidae species involves a vector named as largely unknown. Other disease agents transmitted by the biting midges, including the blood protozoan of the bird, such as the Hemoproteus meleagrides in the turkeys, and leukocytozoan QD in the chicken, and nematode the onchocerciasis in the horses. Biting midges can also cause the discomforting skin reaction in horses known as equine allergic dermatitis, and you see in this picture. Blue tongue disease outbreak occur among the cattle and sheep in the Netherlands, Belgium, France, and Germany in 2006, in Denmark, Luxembourg, Switzerland, United States in 2007. This northern expansion of the blue tongue serotype 8 virus in the northern Europe, attributed in the part to wild-born blue tongue virus infected culicoid females in the Mediterranean to global climate changes. Blue tongue virus 8 was not reported again in Europe until the 2015 in the central France and the United States. The blue tongue virus was first isolated in 1952 in the taxon from the sheep exhibiting a condition known as the sore muzzles, and you observe this in this picture, and it is now known to occur throughout most of the southern and western states, and prevalence of antibody to the blue tongue virus in cattle is currently 20% to 50%. No cases have been reported in the Alaska or Hawaii and Canada, remaining largely blue tongue free. Detection of infected animals was reported there only in the Okanagan Valley of the British Columbia during the localized outbreak in 1975 and 1987, apparently originating from cattle imported from the United States. So the equine onchocerciasis. This equine onchocerciasis caused by the filarial nematodes, the onchocerciasis, widely distributed nematodes transmitted to the domestic animal by biting midges. Horses, only known host, although it occurs worldwide, named that it referred to the infestations or by or onchocerciasis, including the cutaneous equine onchocerciasis, equine ventral midline dermatitis, equine knuckle disease, fistulous wither. Prevalence of onchocerciasis, many region of the United States, up to 85% or more of the old horses infected with this parasite in the New York. And this is a picture of that onchocerciasis. Equine allergic dermatitis, horses exposed to bites of the culicoid species commonly exhibit an allergic skin reactions. Seasonal dermatitis affecting the withers, mane, tail and ears, and back, ventral midline, other body regions, presumably reflecting the feeding sites of the different biting midges, and first attributed to the culicoid bites in Australia in the early 1950s, where it is also known as the Queen's Land itch. Now throughout the world, sweet itch, summer dermatitis, summer recurrent dermatitis, summer eczema, equine culicoid sensitivity, and the Dubai itch or Philippines and Kazan disease in Japan, a similar seasonal dermatitis in response to culicoid bite are also occur in the sheep, and you observe in this in these pictures. Prevention and control, the larvicidal drugs that reduce the population of biting midges, and breeding sites are not easily located and may be so dispersed. Application of the insecticides kill the immature stages. Modification of the habitat reduce breeding sites by filling the living that the areas decaying, regulating water levels or disrupt the breeding and larval development, and eliminating the seepage area or leakage water roughes, and discourage the breeding of the important species, the C. sonorensis and C. variabilis. Proper maintenance of the farm ponds and fluctuation of the water level in dairy ponds and waste lagoons, and reduce the number of adult biting midges. And this is the picture. How can you avoid this? Adulticides use the limited success in suppressing the adults. Effective, they are usually applied as a mist or fogs in the evening hours when the insects are most active. Ultra low volume of formulation of the insecticide in coastal areas, problems, severe area used to salt marshes bordering populated for control of biting individuals, protection of humans and other animals, and discouraging the ceratopogonidae midges from the biting, and scheduling outdoor activities, avoid the peak biting periods of the troublesome species in the advisable, and other prevention and control are animals such as horses can be stabilized at night to protect them from the species that do not rarely enter buildings or other shed. Mesh size of some most windows and door screens like you see in these pictures are not effective in excluding the biting midges, C. fiorensis, C. pariensis that will enter buildings in the search of host, and residual insecticide treatment of resting surfaces such as the walls and roofs of the animal shelter reduce the number of certain culicoid species. Insect repellents applied to exposed skin and diethyl metatoluid that use of the jackets and other clothing impregnated and provide the effective protection from the bites. So thank you so much.